I'm Lutz Froenicke. I'm working at the UC Davis Genome Center, and I'm managing the DNA Technologies core facility, so genomics core at UC Davis. And I'm working in the genomics space for about 20 years. Our long-read sequencing projects at UC Davis are on the DNA space, mostly de novo genome assembly projects. And for RNA sequencing, we see quite a lot of samples coming in for single-cell related cDNA sequencing. So these will be then kinexed, concatenated, and sequenced. These are the predominant types of samples that we are getting and working on. UC Davis is a place where you can try things. It seems to be somewhat our specialty for short-read sequencing as well for long-read sequencing, that people sending us their difficult samples. That means we have quite a lot of experience working with somewhat crappy material. And we have the experience, so our staff has been working on long-read sequencing. We have one staff member who's working on long-read sequencing, PacBio sequencing for 12 years, similar in the short-read sequencing lab. So we have extremely experienced staff. And we're open to weird things, to trying weird projects. In our lab, we have one person which is dedicated, which mostly works on high molecular weight DNA isolations. So this is a PhD level person who has lots of experience. So based on what we have done before, he will make an educated guess which of the different DNA isolation protocols is most likely to be successful. But in some cases, it will be trial and error, and we hopefully have enough material from the clients to allow this trial and error. And basically it comes down to 3 protocols. Plants, 2 protocols, and for animals, 1 additional protocol that we are using, and potentially do some slight modifications to these. But yeah, we have a choice, and nanobind is one of them, and then 2 other protocols that we are working with. Very frequently when customers coming to us, they still have some old made decisions based on some old knowledge. For example, clients really want to complement their long-read sequencing data with short-read sequencing data, which is absolutely not necessary at all anymore. Yes, in this case, we can clearly make a statement. You don't need short-read data for a de novo genome assembly for your project anymore. You would actually make your genome assembly worse if you're adding short-read sequencing data. Things like this. So red flags for hi-fi projects really is if people insist, and some customers will insist on doing their own DNA isolations and have not practice in this part, or really will not put the time into figuring out the optimal protocol for us. So in general, our recommendation is, if you want to do this for genome assemblies or hi-fi sequencing, and you only want to do 2 or 3 samples, best leave it to experienced people, because otherwise you spend months optimizing the protocol and might still end up with suboptimal product. If you're planning to do 20, 30 large numbers, of course, you will be putting in the effort and establishing appropriate protocol for your samples. Go ahead. And we have quite a lot of customers who do their own DNA isolations, high molecular weight DNA isolations, for example, for grape sequencing, which is very difficult sample type, and they're getting excellent results out of it. But yes, really, if it's a one-off or two-off, let people with experience do it. We have been using the predecessor of the Amplify protocol, and we're also using the Amplify protocol for some samples. And this is not only for really low input samples, but we're also receiving samples from clients, and they might be in a very, very poor state, or they might have some contaminants in them that are very difficult to remove. And in this case, it sometimes helps to rescue basically these samples by adapter ligation, amplification, and getting good sequenceable DNA out of crappy input. Our first reactions when we heard about the promises of the SPRQ chemistry were we were curious, and we were very, wait and see attitude, because it sounded too good to be true. But so far, basically we switched in late December to the SPRQ chemistry, and we have not gone back to the original chemistry. We had zero problems, and basically the yields of all our projects, as far as we can judge this, very likely are improving dramatically with the SPRQ chemistry. So this was indeed a big step forward. And the other thing that it allows us to do is obviously work with samples that have much lower sample amounts. So for small critters, it is a big help. So what has changed for us since the first PacBio sequencer that we got in our lab? And now is that, now PacBio sequencing, long-read sequencing is routine. It's reliable, and compared to the very first PacBio sequencer I had, it was really only good at that point for one singular project. We had it for 2 years, and it was only good for one singular project to estimating the repeat lengths in Fragile X syndrome. This is what it was used for. Now it's universal, it's routine, and really what makes me excited for our particular location, since we are at UC Davis, which is an ag school, is really the applications for plant breeding. So that would be, the outlook to do breeding scale long-read sequencing at low coverage for plant genomes. The biggest reason at the moment people are doing sequencing for de novo assemblies is actually conservation genomics. And just one example was the big California conservation genomics project, for which we did the sequencing for more than 150 reference quality genome assemblies. This happened in the last 2-3 years. And basically in this case, the de novo genome assemblies are a tool to be able to study the population biology, the foundation. And then it's very, very variable. There are some labs which studying, for example, spider silk for biotechnology application, something very exotic. Then we're sequencing quite a lot of species from Antarctica. People are looking for meaningful ways genomes adapt to super low temperatures and life in weird circumstances, things like this. So it's not for the purpose usually of, we have another reference genome. There is some interesting biology behind basically all of these. The biggest thing, which is a somewhat unsolved question, or is our clients should be a lot more high throughput sequencing, for example, for plant biology, plant population biology, plant breeding, because if they're not doing long-read sequencing, they're missing out on lots of valuable knowledges about their genomes. So obviously, and high-throughput DNA isolations and high-throughput sequencing are feasible for most plants, but it will still require optimizing protocols for specific samples. And in effect, the DNA isolation is the most laborious part of the whole process. So in any case, in a dream world, there would be some more universal protocol for high molecular weight sample prep, especially for plants. At UC Davis, as you might have got the impression, we are working usually for de novo genome assemblies with quite exotic samples. So this requires trial and error, figuring out what works for, mostly regarding the DNA isolation for these species. These are frequently endangered species where you can only get samples of animals that died previously for other reasons and things like this. So not optimal samples. What I would be looking forward would be a project with a big number of simple samples, like 100 cell cultures or 100 blood samples, something easy. That would be nice for a change.